That was the part before the recording. Very well timed. Yeah, after having said that, it's a pleasure to introduce Jonathan again for his final tutorial. Thanks, Jonathan.
I think I quickly can do this on my own here. I'm not destroying anything. Okay. Good morning, everyone. So it's my final presentation, and this won't be a lecture, right? So this, I think what we did over the last 3 lectures was doing very, very basic biologies and principles, which I think now will give you the background to grill me and grill the research we do in our lab, right? So please interrupt me anytime. And so broadly, we are interested in the energetics of biological systems, and you've seen, now it's not working anymore. Now it's working anymore, okay. So you've seen broadly this slide already over the course of this week about these fundamental questions we are interested in the lab, and what we're trying to understand is, you know, how much energy does an embryo or a cell need in order to stay alive, and how much more energy is it using in order to grow or develop over the course of time? And can we understand this in quantitative terms? And then if we do understand this, can we understand potential energetic constraints? And then also on the scales of subcellular systems and molecular systems, we try and of course understand how cellular metabolism transform these nutritional matter and energy into energy currencies and matter the system can use. And then in time and in space, of course, you have very different cellular processes and active cellular processes, which either use energy, matter, or both. And in the lab, we have broadly like 3 projects, maybe 4, but so during my postdoc, I started to work on this developmental energetics, or the energetics and expenditure, budgets, and evolution. Now recently more the evolution of animal embryogenesis, and this work started together in the lab of Carla Neugebauer and Joe Howard. And the second part was also with Pablo. We went to Woods Hole, a similar course like this, and for 2 weeks, and where we developed a little empirical model of some of these heat dissipations, which actually after the course, Joe started to write the paper in the car, right? So these courses can actually also lead to some new research and some new insights, which in the end will result in a publication. And then I'm working with Pablo more on the thermodynamic constraints of unicellular life. And I believe, Pablo, you're going to talk about this later on. Yeah. And then what we're trying to do is, of course, on the scale of embryos, we try to link whatever we find down to the cellular metabolism of these cellular processes, thinking about how things happen at the subcellular scale. And this is a project of the first PhD student, Anna, together with Sophia, and then the theory was done, F.A. Ilke, who was an independent postdoc in the laboratory of Frank Jülicher at the Max Planck Institute for Complex Systems, Physics of Complex Systems. So these are the 2 projects I'm going to go into today, and conceptually the way we think about it, right? Like if you think about how a cell or an embryo obtains energy and matter from its environment or from stored energy in the yolk, it uses these substrates and then uses classical, what we learn, catabolic reactions to transform these nutritional energies into energy currencies such as ATP and reducing equivalents, and then use these energy and the matter to drive anabolic building up reactions and build embryonic structure, but on the same time also drive all these active cellular processes such as mitosis, you know, chromosome segregation, translation, transcription, and so on. And conceptually, the way we think about it is we think there's a zygote, you know, fertilized oocyte, which then uses these substrates stored in the yolk as a chemical reaction, and over time builds embryonic structure and metabolic products. And this is, of course, characterized by some classical metabolic parameters, such as how much oxygen has been used, how much ATP, but also by some thermodynamic parameters, such as how much free energy has been dissipated, or enthalpy or entropy. And the model system we are using is, you saw in this movie, but this is the depicted schematic of early zebrafish development. And it starts from a 1-cell stage, where you have the yolk and you have the cell on top. And in the beginning, they are not physically separated. There's no membrane. However, there is a density difference between the cytoplasmic and the yolk. So you have this cytoplasmic streaming, and then this embryo divides synchronously over time. And of course, we've learned a lot about in the last decades about how differential cellular behavior, such as cell divisions, differentiation, growth, patterning, gives rise or gives to the emergence of organized structures such as tissues and organs. And that's the classical view of developmental biology. And we understand a lot, but we don't understand so much yet about these energy flows in these systems. And what I did as my postdoc, I started to focus on these early cleavage divisions, right? These rapid, exponentially increasing numbers of cells. Although the overall embryonic volume is constant, meaning that you get exponentially more cells, which become exponentially smaller. And they divide roughly in zebrafish every 17, 18 minutes, or about 10 times or 9 times. And it's a beautiful system because you kind of separate this process of doubling your biomass from the process of just dividing and proliferating. And these early divisions are solely driven by this minimal cell cycle we had, solely undergoing DNA replication during S phase and then chromosome segregation and cytokinesis during M phase. And if you think about an embryo, you can start to think about an allergy balance around the system. So at one point, you have all the nutrients for 5 days of development or 6 days of development stored in this yolk. And this energy now is then transformed by chemical reactions, what we learned as of metabolic reactions in the presence of oxygen to CO2. And you harness that energy by the stepwise oxidation of your nutrients in order to drive or fuel one cell division, for example. And these divisions are, of course, characterized by these parameters. And usually life is exothermic. And so all these reactions also dissipate heat. And this heat, the law of Hess tells us that this heat at constant pressure is equivalent to the enthalpy change of every chemical reaction times their rate. So you can think about the enthalpy entering the system from the yolk, enthalpy leaving the system in terms of these molecules over here. Part of that enthalpy is stored, maybe, in biomass, and the difference is dissipated as heat. The size of the embryo stays exactly. The density of the cells of the embryo, roughly, right? It's a good approximation. There might be something subtle to it if you look in the cytoplasm, and maybe I'll get to it. The cytoplasm at one point seems to get less dense, but that's very preliminary data.
So what are the speeds and those terms in that equation? Tesla.
Sorry, can you speak up a little bit?
That equation,
yeah.
Q dot, yeah. What are the terms in there?
So this is the flux of a certain molecule I. So imagine you have glucose in your yolk, right? And that is taken up by the cell, right? Like, so this is this flux, and this is the enthalpy of formation this molecule takes, okay? And now this goes into the cell, and you can have more molecule, not only glucose, you could have lipids, something else. And that's the reason why you sum this. And then you have something which leaves the system with a flux as well. So there could be the production rate of CO2. And CO2 also has an enthalpy of formation. Right? And so you sum this up. Basically, you look how much enthalpy comes in and how much enthalpy comes out times their respective fluxes. Right? And then you want to stay, maybe some enthalpy stays in biomass. And that's a function of the rate of your growth. And then there is a difference, and that is usually the difference is the enthalpy change of this whole system, and that is usually dissipated to the environment in form of heat, And that's the enthalpic part of the overall free energy change of that. Yeah, there's minus sign, yeah. There's like it's taken, and then the, yeah. And then this other thing you kick out. If you think about, it's not really, CO2 is also product of catabolism, but this is usually what you would say is the anabolism.
Jonathan, I have a question.
Yes.
So initially, when the cells are dividing,
yeah.
they sort of maintain a size or a specific volume, right? And then they sort of start growing in size, and then the entire organism develops. So I was wondering what exactly, influences this. What's the point where the system understands that now I am going to grow out of size and then develop into a whole organism? But what is that? Is there anything known?
You're right. You have a long time, you have this reductive division. You start out with this massive, big cell, right, like the zygote. And then you go reductive to reductive to reductive to reductive divisions. And eventually, you cannot get smaller, right? And then, or like smaller as a cell size. And then eventually, you need to now go to make another cell to make you double your cell mass, and then you divide it. How does this decided? When does this happen is not known. At least I don't know. Right? And there's also the question you could think about, maybe there's a minimal cell size you can achieve, which then triggers.
I was just wondering from a developmental perspective. I'm not at all aware, so I was just thinking. Yeah.
Well, it's an interesting question, right? Like we asked these very simple questions, and I was in the same situation as you. I wanted to know, when does an embryo actually grow in its volume? Because in the beginning, you have this constant volume, and you divide it, you go down, right? And eventually, when does the overall volume grow? And that could give you an idea when this is happening. But this information is a simple question that's very hard to find. And if you think about Drosophila, right, it's in an eggshell. There is no necessarily growth until it reaches this larva. And what we went through yesterday, of course, we can measure this heat, and we can measure this heat using calorimetry in order to measure that. And this is what I started in my postdoc when I was at Yale. I was thinking, can we use calorimetry in order to understand the overall energy expenditure in terms of enthalpy while this embryo undergoes cleavage stage development? And the way I did this experiment, there was an old calorimeter in some facility standing around at Yale, and I thought nobody was using it. So I started using it, and basically this calorimeter works slightly different. It's a differential calorimeter. It's a differential power calorimeter. So what it tries to do is you have an experimental cell, and you have a reference cell and an experimental cell. And if you put something in the experimental cell which produces heat, there's a feedback heater to the whole system. It's like the differential in the temperature is read out, and you cool down your system to be at isothermal temperature. And then you can read out this differential power between the system in order to control this differential. But basically what I did is I staged 30 zebrafish embryos at the 2-cell stage, so we missed 1-cell division. And then I put them in the calorimeter and then see what kind of heat dissipation trajectory you're going to see. And yesterday, you already played around with data from Drosophila, I think from Schuert. And this is what I was in the same situation as you yesterday, right? So you started to measure something, and then you try to learn something from it. And if you do this in zebrafish in the early cleavage stage development, there's a couple of features you're going to see. So this is the data. But basically, what you're going to have is you have an initial heat dissipation rate of roughly about 60, 70 nanowatts per embryo, right? And this is specific because if you do the same experiment with fixed embryos, so you give them paraformaldehyde and you just kill the embryo, and if you cross-link everything, you can see that these guys don't dissipate any heat. And then you can say, okay, what is this 60 nanojoules per embryo? You can convert this into ATP concentrations of ATP per second. If you just give you a little bit of an idea how much ATP is consumed, and that gives you about 25 micromolar of ATP per second, and that roughly corresponds to a cellular turnover of your total ATP pool in this embryo of about 1 per minute. And you can also do this and think about, assume the density is close to water, you assume the volume of the embryo, that gives you roughly a mass-specific heat dissipation rate of about 1 watt per kilogram. Very similar to us at rest. But then, as development proceeds, or this cleavage stage development proceeds, you can see two features of this heat dissipation rate. Once it goes up in the absence of volumetric growth, and it seems to oscillate around this increasing trend. And what you can do is now you can just mathematically decompose this into an increasing trend component, an oscillatory component, and some residual noise. And the first thing which jumped to my mind is these beautiful heat dissipation oscillations, right? Like if you just look at the magnitude, this is roughly about 2% of the mean, but it's very reproducible. And you can see that it nicely oscillates. And this was initially very interesting because you have this very, very synchronous cleavage divisions. We were wondering whether this is really related to the synchronous cell cycle. And one thing you can do is you can count the numbers of oscillations, right? We know that there are 10 synchronous cleavage divisions, but we miss 1 because we stage at the 2-cell stage. So we would expect 9. And if you count it, there should be 9. And on the other hand, if this is linked to the cell cycle, these oscillations will have a similar period as the known cell cycle. And this is also true. These oscillations have a period of about roughly 17 minutes.
The amplitude shows an interesting pattern. Like, why exactly is that?
So we modeled this because we also thought, is this real or is this an experimental design thing? And imagine now we don't have a single embryo in the system, but we have 30 embryos in the system, which we kind of, the best we could, stage them in time. But they're not perfectly synchronized. And over the course of development, they're going to slightly synchronize and maybe come into phase again. Right? So what's going to happen is as it's additive, they go out of phase and come in phase again. And then you get these, these dips in the amplitude.
Okay, but then if I'm guessing that the synchrony between the embryos, will that be similar over multiple experiments, or will?
If you do it always the same experimentally, then yes, right? But it will roughly give you similar trends. So there's some experiments where this is more pronounced, and there's some other experiments where it's less pronounced. Okay.
Right. So when it's developing, the outside heat is not affecting it to a larger degree.
So it's kind of a closed system, but it exchanges gases with the environment. But it's isothermal temperature. Right? So there's no temperature difference while it grows, and it doesn't heat up the media. I think I cannot exclude this, but it would be so little, right? Like if you think about a nanowatt and calculate how much temperature there would be, but it's just it's too fast. It's dissipated to the thermal bath.
Is there a hard cutoff between these 10 synchronous divisions and then the asynchronous ones, or is it kind of a smooth transition?
So it is kind of a hard cutoff. So if you measure this longer, basically the oscillations go away. And that, of course, depends then on the sensitivity of the system. Maybe there's still small ones. But what happens in the embryo is that you have the slight lengthening of the period, and you can already see this in this data if you quantify this. And eventually, the synchrony will fall off, right? And then you have some cell division here, some there. It's longer, a little bit for one cell, another cell. So intrinsically, the embryo becomes asynchronous. And then if you go between embryos, it's even further asynchronous. And then you would lose these oscillations.
But it sounds like a smooth transition to asynchronicity. Is it not?
It's not smooth. It's rather, synchrony, and then something happens, and we can talk about more how this happens. And then you have individual divisions in an embryo, not all the cells divided.
In the second graph, the y-axis remains the same, but there are 3 panels, right?
The 3 panels,
So
right?
what are, I just missed it probably, but
Okay, so this is the raw data. Well, like this is the data, right? And basically what you can fit is either moving average or something else, right?
okay,
And then you subtract this from the data, right?
I see.
And this is what, like, and then, well, like you exactly. And then you can fit also this function subtracted and you're left with
I
noise.
see, okay.
Okay, are there any more questions? And then of course, this is an overall measurement and we see some oscillations. So now the next step would be, can we use this maybe to try to understand something about the energetic of the cell cycle? And so then you can go and come up with 2 hypotheses. We know the underlying biochemistry of this early cell cycle and say, okay, solely comprised of DNA replication and mitosis. So you can think of maybe what we're measuring here is the dissipation of DNA replication itself. Or on the other hand, maybe it's the dissipation of chromosome segregation of the mitotic processes, or any other biochemical process which may be cycles. So now you're going to design an experiment to block both of these phases, and then see how does the heat dissipation respond. And one experiment you can do in the early zebrafish embryo, specific to early development, is use the drug narcotazole, which acts on the microtubules. And what happens in a narcotazole-treated embryo is that it stops dividing and stops replicating. And you can see this in this image. So these are 4-hour-old embryos, and this is a control embryo. And you can see that the cortical is stained with the cortical actin. So you can see the cortex of the cells, and it's stained with Herx or DAPI to see the nuclei. And what you see here is that after 4 hours, this embryo is nicely cellularized, and it has nicely replicated its DNA. But if you look at a 4-hour treated embryo, which should be at this stage, you can see is that it now arrested between the 2 and the 4 cell stage, but also after 4 hours did not replicate its DNA. And you already see that these nuclei start to fragment and the embryo has a problem. It will eventually die. And you now look at the heat dissipation, you can see that these oscillations are still present. Okay. So these heat dissipation oscillations have nothing to do with the physical processes of DNA replication.
You think change, like does the overall heat change or?
It stays roughly. So I'll come to this after this because it has something to do with the increasing trend. I'll answer this in about 10 minutes. So this was puzzling, right? Like, so we're thinking, you know, if you think about the cell biology, these should be energetically, you know, very demanding processes. However, it seems to not contribute early on, and we can discuss why that may be in a bit. But then to get a little bit of an idea, what you can do is you can start to look at this phase relationship of these oscillations with respect to the phase of the cell cycle. Okay. So what you can do is now you can, what I did was basically I followed these oscillations live at the calorimeter, and I took these embryos out in the increasing phase at the peak, at the decreasing phase, and at the trough. And then I used the morphology of the nuclei as a proxy for the phase of the cell cycle. And if you do this, and in the increasing phase, you can see this nicely round nuclei. We all know how they look like. And this is known as an interphase nucleus. This happens where DNA replications happen. But then what you can see is that it transitions to the peak, is that these nuclei or the DNA seems to start to condense. And then, and this is what we know as the pro-metaphase in mitosis. And then while it goes down, you can see now you form these metaphase, metaplate chromosomes, and then these chromosomes get separated at the trough during anaphase and telophase. So from this, you can say that the increasing phase is likely associated with DNA replication, and the decreasing phase is associated with the mitosis process. You look confused, Shashi?
Yeah, no, how was it?
So basically, this is an old calorimeter. It's not the CHUM calorimeter, right? Like you have this chamber, right? And then you sit in front of the calorimeter and you watch an oscillation happen, right? And from experiment, you wait one oscillation. And then from experience, you know, okay, now it's going to go up, and I roughly know the period. So I wait about 5 minutes, and I'm roughly in here, and I quickly take them out and dump them in PFA. Okay, and then the experiment is done, because you cannot continue to measure heat dissipation anymore. And you do another experiment, and now you follow the oscillation to the peak. You take out the embryo, dump them in PFA, and stain. Okay?
Why I'm asking is because you also mentioned earlier that they may not all.
Yeah, not completely, right? So this is just the idealized, we quantified this. Of course, you have a population, and you're going to have some error bars, right? Okay, and this is very, very reminiscent of the oh, sorry, there was another question, sorry.
Just following up from that same question. So if I understand that they are like different embryos are going to be slightly asynchronous, and your like 0.1 and 0.3 are sort of at a similar heat dissipation thing. So like, would you see, like when you're imaging, would you see at the same heat dissipation level, would you see heterogeneity between your embryos? Like some of them would be at interphase and some of them would be metaphase, but it will be indistinguishable based on the heat dissipation.
So if you just, for example, just without looking at the oscillations, just took something at these corresponding dissipations, you don't know which one it was. It could be either one, but if your embryos are roughly synchronous, you have you have a representation like this because you staged them in time beforehand.
Yeah.
Does that answer? So basically what I do in an experiment is, right, like if you sit and you see the raw data as a function of time, you have your heat dissipation rate, and you sit there and you see something like this happening, something like that, and then you say, no, now I'm going to take them out, right? And then take all these out, put them in PFA, and then look at how the nuclei look like, right? And then, of course, this experiment is done.
Yeah, but then in that very point, is it possible that some of them are in interphase and some of them are in metaphase? Because like whenever you
unlikely, right? Like you're always going to have, because in the beginning, what I'm going to do is under the microscope, I'm looking for embryos which look like this, right?
Okay, got it.
And so I stage them in time. So normally, of course, if you don't do this properly, you will also start to see that the oscillation is not as beautiful, right? And then you're going to start, you have a mixture of population. And there's another control experiment. If you manually desynchronize all your population, you will see an heat dissipation like this, and the oscillations are basically.
So basically, at the start of the experiment, you're making sure that
I'm making sure that they're in time, because otherwise we would not necessarily.
Yeah. Thank you.
Confused here. I mean, you said that when you blocked mitosis with nocodazole, so these oscillations were still there.
Yeah.
So how come? I mean,
I'll come to this and that's the next slide. So this phase relationship is very reminiscent of this biochemical oscillator we briefly discussed, which sets the timing of mitosis in these early embryos. And this is centered around this CDK1 kinase, which binds cyclin, which this cyclin is degraded via double negative and positive feedback loop, leads to the oscillation of the activity of the CDK, of this kinase activity, which then in turn can phosphorylate thousands of different targets in order to regulate the entry into mitosis, and it's counteracted by a phosphatase taking this phosphate off, which then regulates the exit to mitosis. So we were wondering, maybe what we are measuring here is the energy dissipation of this. Right? So if you now use drugs to target different steps in this oscillatory circuitry, now you can see that these oscillations are either strongly damped or even absent. And if you target CDK1 or like this CDC25 or the counteracting phosphatase, now you start to dampen these oscillations. So one way to interpret this data is what we're really measuring here is the energy, the dissipation of the cell cycle oscillator independent of the dissipation of the downstream processes. And the beautiful thing in an embryo is that normally what would happen is if replication or mitosis doesn't happen, there's a negative feedback loop, a checkpoint, which would stop this oscillator in order for the cells to not accumulate damage and then kill itself. But the early embryo lacks these checkpoints. So we would have never figured this out doing this in a normal cell. But then you say, do these numbers even make sense, right? So what we can do is, this is where these estimates become very helpful, is what you can do. You can take this oscillatory amplitude and convert it into a unit of ATP per second using an enthalpy of ATP. Enthalpy of the hydrolysis of it. And then you can do calculations and see what kind of cellular processes would give us similar amplitudes. And does this make sense with our conclusion or interpretation of the data that it has something to do with the cell cycle oscillator? Okay. So what you can do is now think about DNA replication, and we know the chromosome size, or like the genome size of zebrafish. And if we now assume, right, each base pair gives you hydrolyzes in pyrophosphate, it's 2 ATP equivalents. It's a diploid organism. We can say over the course, it has to replicate 1 genome. And then if you divide this total ATP via the cell cycle time, you end up with something of a 0.6 nanomolar of ATP per second, which is order of magnitudes lower, at least for one replication of the genome, lower than the oscillatory amplitude we measure. So at least early on, where you have very little cells, DNA replication doesn't contribute significantly to these oscillations. That's the conclusion. Later on, probably it does, but then we also start to lose these oscillations. You can do this. You have these molecules called histones where the DNA is wrapped around, and you can calculate it either without or with histone translations, or we're still on a lower order of magnitude. You can do the same thing, for example, if you take the total amount of tubulin in an embryo, and you would now polymerize the total amount of tubulin exactly at the time. This will now reach a similar order of magnitude compared to the oscillations. However, we only know a fraction, a very tiny little fraction of tubulin is polymerized to form the spindle of the cellular. So this is also, at least early on, or at the 1024 cell stage where the data is coming from, does not contribute too much, right? But then what you can do is you can look at proteomics data from a different species. This, most of the proteomics comes from Xenopus levis, but it can give you a little bit of an idea, you know, how many targets are getting phosphorylated and using ATP and how many targets get dephosphorylated using ATP. And you can now start to realize if we now phosphorylate and dephosphorylate these targets, now you start to see that these ATP usage start to have a similar order of magnitude within a factor of 2 of the oscillatory amplitude. So chemically speaking, what we think is going to happen here is that what we measured is really the dissipation of the cell cycle oscillator itself. And chemically speaking, it is these continuous phosphorylation and dephosphorylation reactions driven by the CDK1 and its counteracting phosphatase PP2A. Okay? There is a question.
Oh, mechanism of action of the drug, I forgot the name, the initial drug that you had used, which did not show the difference?
Cortisol.
Yeah. So what is the mechanism of action of
it blocks microtubule, like it stalls microtubules, it blocks microtubule polymerization.
Okay, so since it was not, got to do with the phosphorylation of the checkpoint proteins, it did not.
They're not there, right? Like they're exactly not.
Okay, so because of that, the oscillations were not different. But when we halt the checkpoint proteins of phosphorylation, we see that the oscillations do die down.
So like in a normal cell, right? Like if they have checkpoints, these drugs will activate these checkpoint kinases, which then stops the cell cycle oscillator. And then one interpretation would be these oscillations go away. Yes. Okay, good. then Yash started to ask, like, what do these narcotisol drugs do to this increasing trend? And this is stuff we realized early on, is that if you treat these embryos with narcotisol, and this is this purple line down here, you can see these oscillations still present in the raw data. However, the overall heat dissipation rate does not increase. Okay. So we were thinking there's some aspect of cell making more cells which leads to this increase in the heat dissipation. Okay. And one way to think about it is what happens, right? Like, let's just make a very empirical, with a lot of assumptions. You can say that we have a spherical cow, or like a spherical embryo, and the cell is spherical. We know that the total volume is conserved while it undergoes cleavage stage development. And we know that the numbers of cells increases exponentially. And then you can think about how does the surface area increase, right? And you can see if you do these calculations, the surface area doubles every 3 divisions. Okay. So now we were thinking maybe we can fit this increasing trend with a very, very empirical model and look how does this, if there is a surface associated term like a B term, how much does it increase over the course of division? Does it go with the numbers of cells, or does it go with doubles every 3 divisions? And so you can fit this data, and you can see 2 curves here. The blue curve is actually fitted where you constrain your model that it should go with the numbers of cells. And the red one is where it should go with every 3 divisions. And you can really see from the fits that this assumption that it maybe scales with the surface area is a valid assumption. And then you can also leave this parameter tau free and get it from the fits, right? And you can see from the fits you can get these parameters. So we have this initial term volume A, which we think is the term associated to this constant volume. And then we have this surface area term B. So this is the surface area at the 2 cell stage. And then you can see that we know the cell cycle doubling time is roughly about 17 minutes. And you can see that the heat doubling time is roughly 3 times that. So this model says that it's probably consistent with some aspect of having more surface area, which gives you this increase. And I will come back to this because we don't fully understand the mechanism yet, but this is where we're actively working on. But basically now what we can do is we can use these overall heat dissipation measurements and then combine it with perturbation experiments and then some empirical modeling to start to build something like an energy budget of this early cleavage stage development. And what I've showed you is that these tiny little oscillations, we can associate it with this cell cycle oscillator, and probably this increasing part, at least initially, with this increase in the surface. But of course, this leaves a lot of this dissipation unaccounted for. And this is what we're currently trying to do, is try to build an overall budget, trying to understand what are these other processes.
So this basically means that most of the energy that's being dissipated by the cell is used to make lipids. Is that right? Or
no, probably not. So
what does surface mean then?
Yeah. Surface it's probably plasma membrane, and initially we thought, so I don't know if I have the slides here. Anyway, what you can do is what we also did together with Pablo and the students is we use this term and see, what do you need to, how much ATP we could convert this into ATP, and then how much ATP would you need in order to make all the lipids for a squared micron of membrane, or make all the proteins, and usually the embryo has a lot of lipids. So we were thinking that, the only thing, what are you going to do is take a head group off and put a different head group on, and it's very cheap. And also thinking all the embryo has all the amino acids, and then what is the, basically of a squared micron, you have 60% lipids, 40% protein. And then you can calculate how much does it take to build this protein, and how much does it take to make this lipids. And if you think they have all the amino acids, synthesis doesn't account for it, or it's orders of magnitude lower. what you then can do is, what if all the plasma membrane ATPases consume a certain amount of ATP in order to keep your membrane potential? And then we had to bound this by a lower and an upper bound, because the in vitro data was basically by a factor of 1,000 different. So you can put a lower bound and an upper bound, and then an upper bound would roughly account for that. So one interpretation is it could be the ATPases in your plasma membrane. We think, depending on how the time is, we think currently it has contributions also from cortical mitochondria. Okay. So this is where I roughly started my lab. Now we moved on a little bit to try, Vashek, and just like a little bit of a side project is, if we write, right, there is this classical system where a lot of these biochemistry of the cell cycle oscillator has been figured out, and this is what we call the Xenopus extracts. Okay. And what you can do is you can take oocytes from a frog, centrifuge them, and isolated cytoplasm. And the cytoplasm, if you give it ATP, will cycle, irrespectively of whether you have a nucleus. This oscillator is going to run. So if we write, we should also see that this extract giving us oscillations in the heat dissipation. And this is just very recent data because they're not easy to handle. What you can see is if you take some cycling extracts, you can also see nicely these oscillations in the heat dissipation. But Vashek is now recently moving on, is working more on mammalian systems, and he was thinking, can we also see heat dissipation oscillations as a function of the mammalian, the classical mammalian cell cycle? And what you can see here is if you block HeLa cells using a drug at the G1 to S transition and release them, and then measure the heat dissipation as a function of time of a synchronous population, you can now see that there's this massive increase in the heat dissipation. So these are 3 different densities of cells with 2 different kind of colorimeters. That's the reason why the noise is a little lower in the upper one. What you can see is that it increases during S phase, and there's some interesting dynamics associated with this G2 M phase transition. But he recently moved on to think what Pablo is going to show you, to try to quantify the energetics of the input and outputs of exponentially growing human cancer cell lines, in order to build an energy balance around the system in terms of how much free energy comes in, how much free energy comes out, and how much free energy is conserved in the system. And he uses calorimetry and then quantitative phase microscopy to measure the mass, oxygen consumption, and all the techniques we actually discussed he's using to try to build this energy balance around a human cell. But what we mainly focused on in the beginning was, now in a collaboration with Pavel Tomanchuk, he's an evolutionary developmental biologist, is trying to now look at these developmental energetics more across the animal evolution. So you can see that these cleavage stage modes are very, very diverse. And across animal evolution, we ask these simple questions: are the energy expenditures evolutionary conserved or divergent across the animal phylogenetic tree? And what Vinka did is, Vinka is our zookeeper. So she's the PhD student running this project, and she has a lot of different kind of beautiful aquatic animal species. So what we have is zebrafish in the lab, and then I don't know if any one of you looked at this Platinerus dromeruli data. So we have this marine annelid worm Platinus maleruli, and then we have Cnidaria, Nematostella, and then we have a sea star embryos. Right? And they're all very different sizes of embryos, and they undergo this cleavage stage development. And you don't have to remember all these kind of cleavage types or gastrulation types, but they're different. They're using different developmental modes, and they have very different egg sizes. And we also have Drosophila and then some sea urchins. Now what Winka did was, initially she started just to measure the heat dissipation rate of these embryos undergoing development for the first 24 or sometimes up to 48 hours. So you can see the data for zebrafish now. We focused on these oscillations that are hidden down here, but now we measure for 24 hours, or we can even measure for days. And then we can do this for Nematostella, Drosophila, Platinerus, and sea star. And qualitatively, you can see that they're all increasing, right? But so the question is, how do you now compare apple and oranges, or fishes and Platinerus, right? And if you plot them all on the same plot, you can see that the magnitude of the dissipation, of course, is very different. And we also know that they all have their optimal temperatures where they grow, so they've all been grown at a different temperature, and you know that every chemical reaction is temperature dependent.
Do these 5 species share common phylotypic stages?
They do, right? Like yeah.
They do. In which stage can you see?
Much later, somewhere.
Oh, okay. So it's a kind of relatively initial stage.
Exactly. It's initial. It's roughly they go cleavage, gastrulation, and then you catch a little bit of the oogenesis.
Thank you.
Can you? Yeah, I was just wondering that the trend in which this is increasing, the graphs are increasing, does it also follow the classification in which they have actually evolved? As in the next graph, the next image. Yeah. So here if you see very high school zoology that I can recall right now is probably Echinodermata. And then, I mean, the zebrafish is the highest phylum that comes up here.
I don't know, we haven't really looked at, first we currently want to do quantitative data, but like the beautiful thing is, do we find a parameter to make a phylogenetic tree independent, just an energetic parameter, right? That would be the most beautiful outcome, right? And this is all preliminary, right? Like it's something which is going on. There will be no answers, you know, like there's not a finishing project in here yet. It's getting there, but
No, I guess it's very interesting. Just the next graph, if you show the next one. Yeah, I think this is the order in which it actually comes up. I think Drosophila belongs to Arthropoda. It's the insect family. And then that will come to Pisces, I think, the fishes. So they are, I think, above or below.
if you can have a look here, right?
Yeah. And then Echinodermata and all are at the bottom.
Maybe, I don't know.
Yeah, so they actually follow the trend in which the animal kingdom has evolved, and accordingly, probably their heat dissipation flow of the embryo is also.
Something to think of, right? Like I think it's a little early to make strong statements, right? That's good. Anyway, so what Winker then thought is that, you know, they're all grown at different temperatures. They have different different developmental times and rates. But she thought there's this interesting observation which comes from the heat dissipation of a frog embryo undergoing development. And we also see this for the amplitude of the oscillations is if you put an embryo at different temperatures, right, it's going to speed up. In here, for example, in the frog data, what you're going to see is that the developmental time speeds up, but also the amplitude of your heat dissipation is increasing. Right? And similarly, if you cool down, it slows down, and also the amplitude of the dissipation is going to go down. But if you look at the integral, right, the area under the curve, or the total heat dissipated over the course of the whole development, you can see that the area under the curve is the same. Right? So the total dissipation for development is independent of temperature. And we see the same thing for the oscillatory amplitude. If we do this at 3 different temperatures, of course, the amplitude and the time is going to scale, but the total area under the curve is the same. Okay, so what Winker then did is, what she went is she looked at the timing of gastrulation and cleavage in all these species, and then looked at the total heat dissipated per cleavage or gastrulation in these different species. And then she plotted this data against the size of these eggs. Okay? So in the next graph, what you're going to see is it's all preliminary data. As you can see on the x-axis on a log scale, you can see that this is the embryonic volume. And then on the y-axis is the total heat dissipated of these embryo undergoing cleavage stage or gastrulation. And on the lower plots, you can see is that is the volume specific total heat dissipated against the volume in a log-log plot. What you can see is that there seems to be a linear relationship, at least for the total energy used in cleavage and gastrulation with volume, or it's invariant if you use the specific. We don't know what, we have too little data to do a real fit and what the exponent is, but looking at this, it looks like the exponent in this one is 0, so maybe there's proportional scaling between the total energy dissipated and the volume. Right? Now the temperature will have the same volume, right? So these guys are all, they all have their different temperature optima, right? So if you look at zebrafish grows at 28.5 degrees, and then these guys grow at 16 degrees. It's cross organisms, right? And then we know when they do what, right? Like cleavage goes from 1 hour to 5 hours, for example, and then you integrate your total heat from 1 to 5 hours, right? And then you plot this heat against the initial egg volume. It's not normalized to temperature per se, but we thought that this total integral may normalize for temperature, but we have to do these experiments a little bit more carefully. For frog, this is the Nagano paper. Yeah. But the point is that this is acute changes of temperature, right? It's not an organism which has adapted to a different temperature range for the course of thousands of years, right? This is an embryo which has been grown. The parent has laid eggs, for example, at 24 degrees. And you take this egg and you put the same eggs at different temperatures, right? No, over the course of development later, for example, if Drosophila develops during the larval stages where there is growth, there is a clear, interesting observation that at lower temperatures, the larva is bigger before it pupates, and at higher temperature, it's smaller. Right? This is not the case in embryogenesis where there is no growth. This is not observed. There's no growth, yeah. Very little growth. Here, there is growth at one point. So anyway, this is preliminary data, but it looks like that it's maybe a proportional scaling, right? So it's interesting that no matter who you are and how you're going to do, but the way we interpret it at the moment is no matter who you are, or I'll finish the sentence short and then no matter who you are, what kind of developmental modes you use to do cleavage or gastrulation, the total energy you need to expend is just a function of how big you are.
Probably didn't get it. So you were saying that you didn't normalize for temperature when you plotted these?
Well, we think that that the total heat dissipated,
right,
per stage is independent of temperature. We currently just thought that we can use the total heat to look at something which has been grown at different. But of course, in the end, in order to say that this is a valid assumption, we should do the experiment that the total heat in each of these individual species doesn't change with temperature. Right? But of course, this is very qualitative. And so we were thinking maybe we can do a little bit more, develop a little bit more quantitative intuition about this data. And we were thinking, this is what you guys did, is can we now not plot the heat dissipation as a function of time, but plot the heat dissipation as a function of any morphometric parameter. And one morphometric parameter what we used is the numbers of cells. Right? And we did that currently for 2 different species. One is the zebrafish species, and the other one is the Platinivorous dromeruli. And maybe we can add a 3rd species if you analyze the Drosophila data. right? Because there we don't know, but we have the data which you would. So what you can see is here, if you now convert time into numbers of cells based on data of numbers of cells against time, and then you use this data to convert time into numbers of cells, and you plot the heat dissipation rate of zebrafish against the numbers of cells. And similarly, the heat dissipation rate of Platinoris against the numbers of cells in the log-log plot. So you can see that there's an interesting relationship between the heat dissipation rate against the numbers of cells, and there seem to be two scaling regimes, one early and one late, in both of these species. So what then Effe did, and he started to collaborate with us, he developed a broad description, empirical description of this relationship. We were thinking that you can write down the heat dissipation rate as something as the heat dissipation rate which is proportional to the initial volume or the numbers of, and then something which scales with the surface area based on this previous results which should increase double every 3 divisions. And this exponent actually here of this scaling regime is roughly 1/3. And then something which is related to the numbers of cells, which switches at a particular cell number and then overtakes. And this should then scale with the numbers of cells, and this is actually roughly true. Okay. And what is really interesting is that these species are very, very different. They do very, very different cleavage modes, but overall they seem to have 2 similar scaling regimes.
Question?
Yeah.
Here. So I was wondering, how would you do to make the same experiment, but with the Drosophila, considering that there is like a late processes of cellularization?
So we don't know, right? Like maybe it's not surface, right? And it's probably a combination of surface with something else. And that's exactly why, I don't know if you realize, but Drosophila is always a little the outlier in these plots, right? Maybe there's something interesting about being a syncytial species. And maybe it's not the numbers of cells, but you can also interpret this N as the number of nuclei, right? It's just a model, it's probably wrong, right? But it gives you a little bit of an intention or like an intuition how to think about this.
Jonathan, I had a question. So if you look at the first graph, it's sort of,
this one.
It sort of shows that there's a saturation behavior that comes about after, I think, around 5,000 into 10 to the power 4 cells. But the saturation behavior is not noticed for the second organism. Also because I think the number of cells are less.
At the moment, yeah.
Yeah, so would you like to comment on this behavior, as in why the saturation and why not the saturation exponential? Is it merely because of the number of cells, or it's something to do with the organism development per se, that they are not saturating, the heat dissipation does not saturate after a specific number or so on?
Short answer is we don't know, right? And for Platinoris, we have more data, but this is still in the making, so we do not know whether it saturates. Maybe there's a third regime, but really no idea.
One question. So all of these animals have external development, right? And
yeah, they're all external, right? Yeah.
So if you were to take a, let's say, thermogenic, warm-blooded animal, do you expect the trends to be different? Because fundamentally it's phosphorylation, dephosphorylation, so you can argue that it can be similar, but they also have a requirement to maintain.
So here, you're right, these are all external developers, and that has a simple reason: they're easy to handle, or easier to handle. And of course, here we're not talking about the oscillations anymore, right? Like, so we do not know whether this is really linked to phosphorylation, dephosphorylation reactions. It may be. That's just a global trend. And for us to do these experiments, for example, in a mouse or in a mouse embryo, is very hard because the embryo is so small. We would need 100 or 200 mouse embryos to do the same experiment, which is, the question is, do we really need it for now? Right? And of course, now the mouse embryo, needs this 37 degrees, and the embryo probably is not able to regulate its own temperature. So but eventually, right, in general, you think about heat dissipation and your uncoupling of your mitochondria, that's a mechanism how you can dissipate all your gradient to do heat and heat yourself up. And this is, of course, also one potential function of this dissipation. Okay. I'm just gonna skip this for now. And then we were thinking now what Anna is doing, trying to relate these scaling regimes. Can we understand it more from thinking about intracellular or subcellular mechanistic understanding of this? And there's something interesting. So if you look at the scaling regime, and this is now all done in zebrafish, if you now look at the scaling regimes and what we initially did, we just sampled something early at the 2-cell stage and something later after, again, these two scaling regimes, and looked kind of like what kind of metabolites are changing. And what you can see is if you compare 4-cell stage embryo against a 2000-cell stage embryo, just relatively, what you can see is that they are definitely not at steady state, right? Like they're charging up the ATP to ADP ratio. So somehow they start becoming more energized. And however, there's something really interesting if we look at these levels of the metabolite pyruvate or oxaloacetate. They roughly increase about 8-fold in their steady state levels between a 2-cell and a 4-cell and a 2000-cell stage. And if we think about, if there's something related to this increase in surface area, which should double every 3 divisions, we would expect that something which is maybe related to this term should roughly increase about 7 to 8. So there's a correlation between the increase in these two metabolites and this scaling regime. And these metabolites are important in your mitochondria. Pyruvate, is imported into your mitochondria, going into the TCA cycle, and oxaloacetate is a TCA cycle intermediate. And what we do know is that if we inhibit mitochondrial function in the early embryo, is you can now start to see if this is your oscillations and your heat dissipation goes up. If you now inhibit the electron transport chain, you can see now that this heat dissipation rate drops to roughly about 50% and the embryo arrests development. So energy expenditure is dependent on this electron transport chain. And similarly, if you measure the oxygen consumption rate of these embryos, you can see it also goes up over the course of early development. And if you inhibit the ATP synthase, also the oxygen consumption roughly drops. So mitochondrial function is increasing during early development, energy expenditure is increasing, and development relies on it. So there is something to the mitochondria. But there's a problem. We know that mitochondrial energy expenditure and function is increasing, but what is known in early development that your mitochondrial content is roughly constant. Right? So this arises like, how do mitochondria meet this increasing demand in time and in space, and how is this mitochondrial activity maybe related to the scaling regimes we see in the zebrafish embryo of the heat against the numbers of cells. So this is what Anna started to think, maybe there is a spatial component to this mitochondria. Maybe not all the mitochondria are active, okay? And so what Anna then did is, if you think about a mitochondria, what we have is a transgenic zebrafish line, which expresses a green fluorescent protein, which has a little targeted sequence, which puts this green fluorescent protein in the matrix into the mitochondria. So what we can see is that we can basically use this line to see all the mitochondria. And then what we can use is a chemical dye, which gets incorporated into the mitochondria based on the membrane potential. And this is what Jingbo alluded to as well. And then what Anna did is she used a dipping lens and imaged the zebrafish from the top and see where do these mitochondria have a potential. So what you can see is initially, if you look at an embryo and just look where the mitochondria are, they are these tiny fragmented little dots, and they are roughly equally, so this is an embryonic cell here, and they are roughly equally distributed, and maybe there seems to be some perinuclear organization of the mitochondria. If you now look at where do these mitochondria have a measurable membrane potential? And you can see that now only the cortical mitochondria seem to have a measurable membrane potential. Of course, you could argue, right, like this is a dye, you throw it out from the outside, it's diffusion limited. You can also inject the dye into the into the embryo. And you can see also nicely now that only the cortical mitochondria seem to have a measurable membrane. And similarly, if you now uncouple the membrane potential, there's basically no doubt. So what you can see is in the scaling regime of the heat with the numbers of cells, when the surface area, you can see this is related to the cortical activity of these mitochondria. Of course, then we can quantify this process, and then this is where Anna collaborated with Marcelo from Robert Haase, and what they developed is basically a segmentation algorithm of these embryonic cells. And then basically in each segmented, they have these concentric rings moving from the periphery into the embryo, and then you can quantify the average intensity in each of these rings and then plot it as a function of distance from the cortex. And what they then both did is they did it over the course of early cleavage stage development, going from the 8-cell stage to the 1,000-cell stage, and then quantified this mitochondrial activity from the cortex of the cells to the nucleus. And what you can see is that in the early embryos seems to be that there's an exponentially decaying normalized signal in this activity going from the cortex to the nucleus. And it looks like that there's an intracellular gradient of mitochondrial activity, similar to a morphogen gradient, but just now in cells. And seems to be that this gradient seems to flatten out. And you can really see these cells become really in their much, and eventually you have no gradient. So maybe that's a beautiful mechanism how you can slowly reactivate your mitochondrial pool in the response to your energetic demands of early development. And then Anna started to collaborate with Effe because an exponentially decaying signal looks like maybe this is a reaction, is consistent with the reaction diffusion system. And then what Effe did is he developed a 3D diffusion model of this mitochondrial membrane potential, thinking about there may be something which sets this gradient, and this is a fixed concentration on the outside, which is then diffusing in and setting this gradient. And then Effe can solve this, this diffusion model in 3D, and we can now normalize the signal, and it's characterized by this characteristic length scale, lambda, right, which sets this length scale of this gradient. And we can now then use this model to fit the data, right, the data we have, in order to think about, can we determine this lambda as a function of developmental time? And you can see that this model, at least early on in development, fits pretty well. And then there's some, it's maybe also a reasonable assumption later on.
Can we go back to the slide where you showed the 3 images on the mitochondria goes to the cortical?
This one?
No, a little bit back. The last slide, I think, before this. Yeah, this one.
Ah, shit.
Yeah, this one. So I was just trying to understand, it's very interesting to see that, yeah, it is going towards the cortical, but what exactly may be happening? The mitochondria is, sort of translocating to the periphery, or it is not, I mean, it is there at its location, but it's not
so they wiggle around.
Functional, or what exactly?
So they barely move, right? Like if you just look at a higher resolution with a higher time resolution and spatial resolution, this mitochondria just wiggle around. There's no movement, and they're roughly evenly distributed in these cells, right? Like if you look at a higher image, you can see these tiny little dots of mitochondria. So there is no, all the mitochondria everywhere. So somehow in the periphery, there is something which the mitochondria can use in order to charge up their potential. And this is what we think currently is maybe a reaction diffusion system, and this is something which we do not know what it is yet, set by the C out, which diffuses in and then is used by these mitochondria with a certain degradation rate K.
Okay, I want to know maybe at which level of temperature we are measure this.
So 28.5 degrees Celsius.
Sorry?
28.5 degrees Celsius.
28.5.
Yeah.
And why you don't, maybe when you scale the models, you don't introduce this temperature for the scaling mode, for the scaling reaction diffusions equations of the consecration?
We haven't done any temperature, right? Like the diffusion coefficient, of course, is temperature dependent, but we don't incorporate temperature right now.
Yeah, because I was thinking that it's possible to introduce the temperature and then the fit between the experimental and theoretical expression can be more fitting a little bit. Maybe it's one of the ideas I have.
So at one point, we were discussing temperature because it could give us a little bit of an idea what this kind of molecule could be, right? Because, diffusion, if it's purely diffusion, right, like, and this diffusion coefficient is going to change the gradient, and the decay rate is going to be differently dependent on temperature than D. Right? And maybe that gives you a little bit of an idea that what kind of molecule it is. We don't know yet what this molecule is.
Okay, thank you.
Something similar to what Pavel was saying. So also as development goes on and the size of the cell changes, if the shape of the cell changes as well, that cinch function will change.
Yeah.
And we know that if it's 3D, it's cinch, but if it's 2D or it becomes flatter, it should become linear. Or
so maybe there is they look very spherical, and we assume it's a sphere, right? But we haven't really looked beautifully at the morphology yet. But maybe you're right. This more linear could be like a more squishing of the cells.
Exactly.
And,
if one dimension is small,
that's a good
then the
point.
others will-
Like maybe we should look into this.
Can you just quickly show us, quickly explain the segmentation slide?
Okay. So basically, this is the top view of an embryo, right? And we take, a Z slice of that embryo, right? So what you can see here, these green dots, tiny little dots, are the mitochondria. This early embryos is full of mitochondria. Like they are not a network. What you can see is, and then they have, if you look at the copy number of mitochondrial DNA, they have roughly 60 million copies of mitochondrial DNA. Because there's no mitochondrial replication, until about 48 hours later, right? So they inherit all their mitochondrial DNA copy number until 48 hours, and then that's maybe a good sign when do they actually start to grow. And then what you can do is you can try to segment these boundaries of the cells, right? And then you overlay a mask. And then you can use this mask in order to define coordinates and define these concentric rings on the boundary of these masks. And these concentric rings, of course, have an area. And then you can use this area as a mask to say what is the average intensity in this area. And this is your one data point, and of course, this is going to give you some distance. And then you do this for the next ring and the next ring and the next ring. And then you can normalize the signal, right? And then put the highest one, and this is what he does as a function of the distance. So the width of the rings is then translated into distance based on the pixel size.
Thank you.
And then what you can then solve for is this decay length lambda, right? Where you can calculate that, and you can see that if you plot that as the numbers of cells, is that the lambda is roughly constant, right? Roughly about 15 to 20 micrometers. And this comes from only the mitochondrial signal. And this two data is depending what width size you set of your concentric rings, right? It's independent of that. But you can also do this for the density. So what you can do is you can normalize this signal of this activity or this potential to the signal of the mitochondria. Right? So you have a density, and you can see a similar decay length of roughly 15 to 20 microns. Okay, and then now the question is, of course, it's a reaction diffusion model. The question is, can we now tune this length scale? And we cannot at the moment. But what we're trying to figure out is, of course, what is that molecule diffusing with that length scale setting this mitochondrial potential? And of course, we're currently thinking, what is mitochondria using in order to charge up their potential? Is it pyruvate? Is it lipids? Is it amino acid? Is it something else? Right? And currently we're doing a drug screen, but the model can already infer you because we know what this lambda is, so we know what this ratio is, and we can take known diffusion coefficients and see what kind of the degradation rate should it be. This is the 15 micron line. And what kind of degradation time should it be if it would be oxygen, NADH, or proteins? And oxygen would be up here. So it's probably not oxygen setting this gradient. And for the others, we do not know yet. But of course, you can then say, is this enough? Is this gradient enough in order to slowly activate your mitochondrial fraction while the embryo undergoes development?
Sorry, I missed the argument in this slide. So what is it that you don't know, the degradation time of this?
No, so we know the diffusion coefficients, right?
Yeah,
and this is the line which gives you the 15 microns, this ratio of D to K. Right?
Okay.
And now oxygen has the diffusion rate of this, right?
Yep.
And we can go to the oxygen consumption measurements and look if K in vivo would be somewhere on this line.
Okay.
And the K in vivo is somewhere up here.
Okay. But for the other two?
For the other two, we do not know.
You don't know K?
We don't know K.
Okay.
But what we can do is, based on the stoichiometry with oxygen, make assumptions of, for example, if a lipid comes in, we know the stoichiometry of lipid to oxygen. So if it would be a lipid, and then it could, we can try to relate it to the K of the oxygen or infer the K of the lipid based on the oxygen in the mitochondria. But this is a little too early, so we don't know the molecule yet. Could be maybe ADP coming from the surface. And then whatever, is this enough, this gradient enough to account for this gradual reactivation of your mitochondrial activity while embryonic development proceeds? And you can now calculate, based on this length scale of this diffusion coefficient, length scale of this reaction diffusion system, you can calculate the fraction of mitochondria, and you can see that roughly at the 1,000 cell stage, you have roughly activated most of your mitochondria.
You said there are roughly 60 million. How many mitochondria copy? What is the copy number?
60 times 10 to the 6.
60 times 10 to the 6.
Or no. Like I'm mixing this up with the volume, but it's 2 to 10 to the 6.
2 to 10 to the 6.
It's in the millions. The order.
Somewhere of this order. So, but when the cells have exited this stage and then now they are normal differentiated cells, what is the copy number then? Does it increase or it stays the same?
So per cell later on when replication starts, so of course it's not measured individual, but it's an average over the whole embryo. It's roughly 30 copy
numbers. I didn't get it, sorry again.
So like it's roughly, if you take a 48-hour embryo, right? And you know how many cells there are and how many mitochondrial copy numbers there are, and then you do an average, it's roughly 30 copy numbers per cell.
30 per cell?
30 per cell, but the cells are much smaller. So maybe the concentration of mitochondrial copy numbers.
The green dots that you were showing in each and every partition, that's a cell, right?
No, the green dots are mitochondria.
In a cell. The boundary represents the cell.
Exactly.
And those green dots in one cell, you're saying, is 2 10 to the power 6.
No, the total.
The total is 2 10 to the power 6. Per cell, it turns out to be 30. But
no, per cell, much later, it's 30.
At much later, it's 30. Okay.
And then you can see, can you try to understand how is this evolution of this related to major developmental events, right? And what you can see is that the radius of the cells, right, like, is inversely with the numbers of cells, right? And then you can estimate, based on this diffusion coefficient, right, when does this gradient reach the nucleus? At what cell stage, right? And can we maybe do this n star when the scaling regimes changed, right? Maybe this is related to this mitochondrial, intracellular mitochondrial activity gradient. And then if you do this for zebrafish and you assume a certain size of the nucleus, a certain size of the initial embryo, or like the radius, and then this lambda, you can see that this lambda eventually, eventually is going to reach and touch the nucleus, right? And this is roughly at the 400 to 600 based on this model. And this coincides with a major developmental event known as zygotic genome activation. This is when the embryo becomes self-sufficient and activates its transcription in the nucleus. And if you now combine again the cell number data with the heat data, this is roughly when the scaling regime changes in the system. If you now look what's going to happen to the mitochondria, right? So what you're going to see here now is roughly at that stage, a bit later in development, you can see these mitochondrial GFP here in this movie and focus maybe on one cell while we image, right? And if you focus on one cell, maybe on this one here, you can see an interface. There's this ring of mitochondria forming around the nucleus, right? So this is a still, and this happens not early in development. This happens only when the mitochondria, which have a membrane potential, touch the nucleus. If you look in electron microscopy, you can see that there's a lot of mitochondria in the periphery to the nuclear membrane, and there's some electron-dense structure between the mitochondria and the nucleus. And if you do this early in development, prior to when the gradient has not reached the nucleus, you can also sometimes see mitochondria in the periphery, but they never form this electron-dense structure. So what we're currently thinking is that maybe this change in the scaling regimes is related to something happening in the nucleus, and maybe this is related to transcription in the nucleus. So what then Effa did is he redefined our previous model based on the data we now have. And you can write down now the heat dissipation rate again as a function of the volume, but now the surface term becomes two terms. It becomes one term which is associated with this energy uses of the plasma membrane, but also something which is related to this cortical activity of your mitochondria as a function of the fraction of the mitochondria which is active, plus something which happens in the nucleus. Exactly, and we can calculate this fraction based on this length scale of this gradient. And basically, this model beautifully fits the data in the domain of the numbers of cells. And of course, you can now go and fit this model to the data and get some numbers for these coefficients. And you can see that we have to, for technical reasons, do this upper and lower bound for the surface term, because otherwise it gets eliminated in the fits. And you can see now we can get a volume term, this QM term, and a QN term for the nucleus. And now the beauty is, again, we can go back to this cost estimates and see whether these numbers make sense. So basically, what we're currently doing is seeing is that these mitochondria are fragmented and evenly distributed in these embryos. And this membrane potential is not homogeneous, but it forms this intracellular metabolic gradient. And it has a characteristic length scale and is conserved throughout embryogenesis. And the cool thing is, if you do this now, not only in zebrafish, but if you look in other species across evolution where we see similar scaling regimes, you can also now nicely see this intracellular gradient in mitochondrial activity. So the way we're currently thinking is you have these cells and you have this gradient, and as these cells reductively divide, this gradient stays constant and slowly activate more and more and more of your mitochondrial activity once it touches the nucleus, right? And this is a beautiful mechanism how you can regulate the amount of metabolic activity in your mitochondria based to your morphology. And we know that at least in mammalians, from a medical point of view, is the mitochondrial activity has to be in this Goldilocks zone. You don't want to be too active because you're going to produce reactive oxygen species, you damage your genome, but you also don't want it too little active because then you cannot drive embryonic development. And if you couple it to your morphology, that would be a nice way how you can slowly reactivate it. And then, of course, maybe there is something happening when these scaling regimes happen, and maybe already has a function saying, once it touches the nucleus, maybe it provides some additional metabolites for zygotic genome activation. That's something we want to currently looking into. And maybe, I don't think I have time to go through the EM stuff, but maybe a little bit. basically, now the question is, of course, what is different between these mitochondria, which have a potential, and the other ones, right? So is there a relationship between mitochondrial function and mitochondrial structure? And they're all little, tiny little fragmented dots. But what we thought is maybe these cortical mitochondria have some different ultrastructure than the other ones. And we don't have an answer yet, but what we did is we did serial block phase electron microscopy. Basically, we took an early and a late embryo, and you do serial sectioning through the embryo, and you do EM. And then what we can do is we can segment these mitochondria, reduce the data, and then have a look at the ultrastructure of these mitochondria in these. And basically, this is a data set of a 1,000 cell stage embryo. And what you're going to see is here, this is the gold phase around it, and this is one cell. And what you can do is you can get an EM image going through the whole embryo. You can see the nucleus appearing here. And now the contrast is going to change. This is just pure raw data. And you see some lipid droplets over here, mitochondria here and there. And you can really scan through the whole cell and have a look at the ultrastructure of this whole cell. Basically, you can also see this interesting nuclear membrane folds. A lot of mitochondria. You have the membrane here. Another cell is appearing over there. Basically now, of course, you need to, one can segment, for example, out the mitochondria or any other ultrastructure and see how that changes in time and in space. And then what a bachelor student did was, basically we developed a synthetic mitochondrial generator in order to validate algorithms which we're going to use then to look at these mitochondria. And can you tell me which ones are fake and which ones are real? There's 3 rows of fake mitochondria. And 3 rows are and 2 rows are real mitochondria. The 3rd row is fake. That's purely fake, like purely elliptic, right? Like, this is clearly fake, right? Yeah? Like first, yeah. The last 3 are fake, right? The other ones are real, right? Anyway, what Paul did is, I don't want to go into the details, but we are really good at faking data now and faking mitochondria. We can use them. We can use them to validate, our algorithms we're going to use, right? And anyway, what we do is we do 2D morphometrics and 3D morphometrics of these mitochondrial structure. The roundness, compactness, surface, volume, sphericity. And then you can, of course, a lot of these parameters are correlated intrinsically. And you can do this. And then you can get a lot of data and basically do a principal component of early versus late. And you can really see that some of these parameters are going to change as a function of developmental time. And we have some indication that this, at least in the early embryo, we haven't done the segmentation yet, but this movement in the PCA is also present in these cortical mitochondria. So they are reminiscent of later mitochondria already. And then I think I'm just gonna stop here. Thank you very much.
Thanks a lot. So we will cut from the recording the part about faking mitochondria.
Yeah, exactly. Cut it.
Questions?
Yeah.
Yes.
When you do this EM for different mitochondria across development,
yeah.
Does the number or the surface area of the crystal change over time?
No. So what also Paul did is, I don't know if it's already in this data set. No. But what he also did is he developed algorithm to get the crystal spacing and the volume of the matrix versus the crystal, and the density of the crystal stays the same. What happens, these mitochondria are just slightly becoming bigger, but the density of the crystal are the same.
In that slide where you had 3 fake rows of mitochondria, how could you identify the mistakes?
Sorry, I didn't, can you just louder?
So you had a slide where there were like 3 rows of fake mitochondria images? Yeah. How could you tell that they were fake? Like
you can tell the difference is very spherical, right? On the one hand, and this is also very round and exactly. At one point it gets really hard, right? Like, to tell that, because the way you do it, right? Like you can make these shapes, and then at one point you get a really cool, you can add noise, and they really look like an EM mitochondria. That's the whole point, right? Like we want to validate or test these algorithms, and then we know exactly what we're going to put in, and then whether the algorithm overestimates a certain parameter or not.
Yes.
The data where there was changing temperature and the time of development was changing. It seems that there's an interesting relationship between time to develop and temperature. Seems like it's inversely.
So lower temperature, it takes longer to.
And yeah, I don't know what your thoughts are on.
Like there's a lot of people right now, because there's a lot of classical developmental biology was always thinking, it's a hot topic. If you can tune time, this is what everyone wants to know. How does an embryo determine time? Right? Like they all do similar things, but very different time scales. And a lot of people now look at the role of metabolism and in developmental timing. But maybe temperature is a way to get a little bit of an idea how you determine time. What I find remarkable, right? We have these millions of chemical reactions. They all have different activation energies, and you change temperature and everything scales proportionally. How is that done? Maybe you can use temperature to learn something about.
Question?
Yeah, so as you showed that slowly the mitochondria were touching the nucleus as the divisions progressed, but the overall volume was the same. So I was just wondering how much of this is is a function of mitochondrial the function of mitochondrial function increasing over divisions, and like one aspect is that, the other aspect is as you create more nuclei and you have more more divisions within the same volume, the nuclei, if you even have the same population that is active, the nuclei, due to the increase in the number of nuclei, you're going to touch it anyway at one point.
How does this? Yeah, like of course eventually, right? Like this is, but because the length scale is the same. Right?
Yeah.
Exactly, and you're absolutely right, right? Like you make reductive divisions, you have a constant volume of the nuclei, and eventually your cytoplasmic to nuclear volume is decreasing, and your nuclear volume to cytoplasm is increasing, and eventually, of course, if you have a constant length scale, that will touch, right?
Yeah. So
exactly.
Yeah. So that's, and it can also be that the mitochondria were uniformly distributed, but their activity was different.
Yeah.
Right. So the activity could also, like the gradient of activity was also decreasing. So could there be two different things? One is the cytoplasmic to nuclear volume ratio. The other one is a loss in the gradient of the activity.
So the point is that the activity gradient is constant, right? So it's not, at least from the lambdas we get from the fits, it suggests that there is no reduction in the activity. However, right, like if this is constant and you imagine you have a nuclei, you have a gradient, there's some distance between the gradient and the nucleus. Now you divide in half and you move closer. And you move closer again, and eventually there's no space anymore. Yeah. This is automatically when we think they have activated most of their mitochondrial pool. And interestingly, there's been a proximity of the nucleus, and it could be, okay, there's nothing. But then we have this preliminary data that there is some active biology happening with the nuclear membrane and the mitochondria, and it coincides with the initial wave of transcription. Right? It's purely correlation at the moment. There's no mechanistic understanding.
So it's largely because of the change in volume metric ratio.
It's probably the whole thing is just because the cell is dividing in half, right? Like that's the underlying. But of course, why is this gradient constant? It doesn't scale with cell size. We know that a lot of morphogen gradients scale with cell size. Right? But it's on the one hand, if this is actively read out, it could be a beautiful ruler, right? To say, what is my cytoplasmic to nuclear volume?
Okay, so if there are more questions, I suggest we can continue the discussion during the coffee break. So let's thank Jonathan again for the very nice course he gave us during this week.